I never really understood the term limited life until I heard about cystic fibrosis. A patient could be diagnosed with CF as early as the age of 2 months, and the lifespan of a CF patient is normally 40 years. And for most of these years, they're probably going to be surrounded by a bunch of medical equipment. And the crazy part is that there isn't even a cure. Every CF patient has a different problem, a different solution, and a different outcome. The best approach for this disease is to have the best treatment. And in order to have the best treatment, it needs to be personalized. Cystic fibrosis is a genetic disease that affects many organs, but mainly the lungs. As air travels down the pipes in the lungs, it interacts with a substance known as mucus. The main role of mucus is to capture dust and germs, so it basically filters out the oxygen as it travels down the airway. A normal person would have mucus that captures dirt, and then cilia would move that mucus out of the lungs and replace it with a new layer of mucus. However, in cystic fibrosis, a patient would have difficulties experiencing this. In CF, the mucus does capture dirt, but the cilia is unable to move that mucus. Therefore, it can turn into bacteria or even block the airways. One of the main causes for this is the CFTR protein. The cystic fibrosis transmembrane conductance regulator, CFTR, is defective in cystic fibrosis. This protein is also a channel that transports different molecules such as chloride and biocarbonates, and it sits on the surface of the cell. In cystic fibrosis, the CFTR protein is encoded by a CFTR gene that is located on chromosome 7. Mutations in this gene could cause cystic fibrosis, and one of the most common mutations is the F508del. The process in making the CFTR protein first begins with CFTR mRNA translation. After translation folding and core glycosylation in the ER, it makes its way to the Golgi complex where it is fully glycosylated. Thereafter, it is exported to the epithelial cells where it acts as a channel to transport chloride and biocarbonates. The CFTR protein is made up of a chain of amino acids that are grouped into 5 functional regions. These regions are known as domains. 2 transmembrane domains, 2 cytoplasmic nucleotide binding domains, and a regulatory domain make up the entire CFTR protein. Each TMD contains 6 segments that completely cross the phospholipid bilayer and allows anions to flow through. The TM segments are connected by 3 extracellular loops and 2 intracellular loops in each TMD. Furthermore, the 4th extracellular loop possesses 2 N-linked glycosylation sites known as N894 and N900, and these are assessed by the ER quality control during the protein folding process. The NBDs and the RDs are exposed to cytosol and are rich in charged residues. These glycans are modified upon transversing in the Golgi complex and may interact with extracellular macromolecules when the protein is located at the PM. The NBDs presents highly conserved sequence for ATP binding and hydrolysis. Meanwhile, RDs are highly disordered and there are multiple consensus sequences containing serines and threonines for phosphorylation for protein kinase. ATP and CAMP-dependent PKC and pKa phosphorylation induces alterations in the CFTR protein conformation, therefore allowing anion conductance through the pore. Among the 48 ATP binding caskets, or ABC transporter family, CFTR, also known as ABCC7, is a very unique one since it possesses an RD and it functions as an anion channel. In cystic fibrosis, there are 3 genes that are deleted. These 3 genes are responsible for producing amino acids known as phenolin, which helps structure the CFTR protein. When these genes are deleted, it results in either a lack of CFTR protein or abnormal functions. For example, a normal person would usually have sweat glands that extrudes sodium, chloride, and water as a hypertonic solution from the epithelial cells. Then, the fluid would be reabsorbed and finally brought to the skin surface, and this fluid is known as sweat. However, in CF, the fluid is extruded, but not reabsorbed, which results in a hypertonic solution being brought to the surface. In secretions, it's a bit different. So this is a diagram of a respiratory secretion. Normally, chloride is secreted, meanwhile, sodium and water is reabsorbed. During CF secretions, chloride becomes trapped in the cell, while sodium and water is significantly increased. The fluid is now isotonic. You can think of it as a domino chain. If one chip falls down, so will the rest. In cystic fibrosis, it starts with 3 deleted genes, then lack of amino acid, resulting in dysfunctional CFTR proteins, and finally, abnormal secretions. Though there isn't a cure for cystic fibrosis, there are a couple of treatments. This includes a CFTR modulator. The CFTR modulator is used to enhance or restore the function and ability of a CFTR protein. This could be classified into 5 groups based on its effects on the mutations. But there are 3 common modulators, and this includes the potentiators, correctors, and amplifiers. The CFTR protein is kind of like a tunnel and has a closed gate. So potentiators are CFTR modulators that keep this gate open and allow chloride to flow through the cell membrane. The drug Avactafor is actually an example of a potentiator. This drug is used to help patients that have gating and conduction mutations in the CFTR. So one of the main 2 problems with the CFTR protein is that it's either not reaching the cell surface or the protein doesn't allow enough chloride to pass through. So by holding the gate of the CFTR protein open, potentiators can allow chloride to flow through, and it can help reduce the symptoms of cystic fibrosis. The next type of CFTR modulator is known as correctors. Correctors help the CFTR protein form the right 3D structure. It could also help it move or traffic to the cell surface. One of the most common reasons on why a CFTR protein doesn't work properly is because of its structure. And by fixing that, we can allow ions to flow through the channel more easily. Nearly 90% of CF patients have at least one copy of the F508del mutation, and this mutation prevents the CFTR protein from forming the right shape. However, corrector drugs are used to help the CFTR protein form the right shape, travel to the cell surface, and stay there longer. But even with correctors, only some of the CFTR proteins reach the cell surface. Additionally, the proteins that do reach the cell surface don't open up the gate to allow the chlorides to pass through. But if the corrector could be combined with the potentiator, it could have the potential to hold the gate open for the CFTR protein. Recently, there was a combination of 2 correctors and 1 potentiator. The 2 correctors were elexacaftor and tezacaftor. Meanwhile, the potentiator was Avactafor. When all these drugs were combined, they resulted in another solution known as Trikafta. Trikafta was a triple combination drug that could be used to treat people with CF who have at least one type of mutation, most commonly the F508del mutation. And the last type of CFTR modulator is known as amplifiers. Amplifiers prevent the amount of CFTR proteins that are produced. In some cases, there are a lot of dysfunctional CFTR proteins, or just in general, a lot of CFTR proteins, and amplifiers are used to limit that amount. So there isn't that big of an explanation for this, but all it does is just prevent the amount of CFTR proteins that are produced. Humankind has made so many discoveries. One of its most significant discoveries is medicine, but the only downside to that is its efficiency. That's why most cystic fibrosis organizations use a method called therapeutic, which provides CFTR modulators to the right patient at the right time. And this could result in saving money, saving time, and providing more accurate treatments. The future of cystic fibrosis is personalized medicine.